A diverse and pluralistic society where every person understands and plays to their strengths, building a fair and self-sustaining model for education rather than knowledge. Universitas Sebelas Maret is here to align Education 4.0 with Industry 4.0. Universitas Sebelas Maret was established on March 11, 1976. As one of the most outstanding universities in Indonesia, Universitas Sebelas Maret has noble ideals to develop the country.
Universitas Sebelas Maret is located in the suburban area of Surakarta, colloquially known as Solo, a city in Central Java, Indonesia. The city is located within a 50-minute flight from Jakarta and 1-hour flight from Bali. Solo is well known as a city of culture and the capital of batik.
Universitas Sebelas Maret Surakarta has grown rapidly over the years. It has positioned itself to become the leading institution in Indonesia. Universitas Sebelas Maret strives to provide high-quality education, develop scientific knowledge and technology products at an international level. With its adequate and appropriate learning infrastructure, Universitas Sebelas Maret offers a unique and engaging learning environment and provides integrated IT facilities and information system in a convenient academic atmosphere and green campus area.
The library features thousands of books and provides access to a wide variety of international journals, which is expected to enhance the learning of the students, faculty, and researchers.
In addition to the fundamental concepts and skills required for a successful career, a graduate of the Universitas Sebelas Maret is armed with soft skills by taking part in various student activities on campus and student organizations both at national and international level.
Students also have the opportunity to acquire global experience through student exchange programs, global internship programs, multilateral research projects, and other international collaborative activities. Universitas Sebelas Maret encourages all researchers to promote their work through publication in national and international journals, as well as other forms of dissemination, such as presentation at national and international scientific forums. Researchers have produced excellent research products and become engaged in the activities of the community through various community service programs.
Active partnership and strong collaborations have become inseparable parts of Universitas Sebelas Maret, without which progress in education, research, and community service wouldn't have been achieved. Our international partnerships create ample opportunities for student and staff exchange, research collaboration, and community service. International collaboration helps to give a global perspective on learning in order to address global challenges while still functioning in a national environment based on potential and local wisdom.
Universitas Sebelas Maret has three main agendas toward a world-class university. Acceleration in various fields of development is needed by embracing the foundation of UNS, Benteng Pancasila. Therefore, 5 strategic policy pillars were decided.
We gladly welcome you to explore and experience our program and to enjoy our campus life, which depicts a blend of cultural power, professional life, and technological advance. Universitas Sebelas Maret, toward a world-class university.
Good grammar and spelling are important, but if you want to write essays that inspire, messages
Students, giving the workforce of tomorrow the tools to become active lifelong learners, independent problem solvers, and create a diverse and pluralistic society where every person understands and plays to their strengths, building a fair and self-sustaining model for education rather than knowledge. Universitas Sebelas Maret is here to align Education 4.0 with Industry 4.0. Universitas Sebelas Maret was established on March 11, 1976. As one of the most outstanding universities in Indonesia, Universitas Sebelas Maret has noble ideals to develop the country.
Universitas Sebelas Maret is located in the suburban area of Surakarta, colloquially known as Solo, a city in Central Java, Indonesia. The city is located within a 50-minute flight from Jakarta and 1-hour flight from Bali. Solo is well known as a city of culture and the capital of batik.
Universitas Sebelas Maret Surakarta has grown rapidly over the years. It has positioned itself to become the leading institution in Indonesia. Universitas Sebelas Maret strives to provide high-quality education, develop scientific knowledge and technology products at an international level. With its adequate and appropriate learning infrastructure, Universitas Sebelas Maret offers a unique and engaging learning environment and provides integrated IT facilities and information system in a convenient academic atmosphere and green campus area.
The library features thousands of books and provides access to a wide variety of international journals, which is expected to enhance the learning of the students, faculty, and researchers.
In addition to the fundamental concepts and skills required for a successful career, a graduate of the Universitas Sebelas Maret is armed with soft skills by taking part in various student activities on campus and student organizations both at national and international level.
Students also have the opportunity to acquire global experience through student exchange programs, global internship programs, multilateral research projects, and other international collaborative activities. Universitas Sebelas Maret encourages their work through publication in national and international journals, as well as other forms of dissemination, such as presentation at national and international scientific forums. Researchers have produced excellent research products and become engaged in the activities of the community through various community service programs.
Active partnership and strong collaborations have become inseparable parts of Universitas Sebelas Maret, without which progress in education, research, and community service wouldn't have been achieved. Our international partnerships create ample opportunities for student and staff. International collaboration helps to give a global perspective on learning in order to address global challenges while still functioning in a national environment based on potential and local wisdom.
Universitas Sebelas Maret has three main agendas toward a world-class university. Acceleration at various fields of development is needed by embracing the foundation of UNS, Benteng Pancasila. Therefore, 5 strategic policy pillars were decided.
We gladly welcome you to explore and experience our program and to enjoy our campus life, which depicts a blend of cultural power, professional life, and technological advance. Universitas Sebelas Maret, toward a world-class university. It's time to bring education into the 4th Industrial Revolution. Flexible, tailor-made curricula are taught by teachers who become mentors to their students, giving the workforce of tomorrow the tools to become active lifelong learners, independent problem solvers, and create a diverse and pluralistic society where every person understands and plays to their strengths, building a fair and self-sustaining model for education rather than knowledge. Universitas Sebelas Maret is here to align Education 4.0 with Industry 4.0. Universitas Sebelas Maret was established on March 11, 1976. As one of the most outstanding universities in Indonesia, Universitas Sebelas Maret has noble ideals to develop the country.
Universitas Sebelas Maret is located in the suburban area of Surakarta, colloquially known as Solo, a city in Central Java, Indonesia. The city is located within a 50-minute flight from Jakarta and 1-hour flight from Bali. Solo is well known as a city of culture and the capital of batik.
Universitas Sebelas Maret Surakarta has grown rapidly over the years. It has positioned itself to become the leading institution in Indonesia. Universitas Sebelas Maret strives to provide high-quality education, develop scientific knowledge and technology products at an international level. With its adequate and appropriate learning infrastructure, Universitas Sebelas Maret offers a unique and engaging learning environment and provides integrated IT facilities and information system in a convenient academic atmosphere and green campus area.
The library features thousands of books and provides access to a wide variety of international journals, which is expected to enhance the learning of the students, faculty, and researchers.
In addition to the fundamental concepts and skills required for a successful career, a graduate of the Universitas Sebelas Maret is armed with soft skills by taking part in various student activities on campus and student organizations both at national and international level.
Students also have the opportunity to acquire global experience through student exchange programs, global internship programs, multilateral research projects, and other international collaborative activities. Universitas Sebelas Maret encourages all researchers to promote their work through publication in national and international journals, as well as other forms of dissemination, such as presentation at national and international scientific forums. Researchers have produced excellent research products and become engaged in the activities of the community through various community service programs.
Active partnership and strong collaborations have become inseparable parts of Universitas Sebelas Maret, without which progress in education, research, and community service wouldn't have been achieved. Our international partnerships create ample opportunities for student and staff exchange, research collaboration, and community service. International collaboration helps to give a global perspective on learning in order to address global challenges while still functioning in a national environment based on potential and local wisdom.
Universitas Sebelas Maret has three main agendas toward a world-class university. Acceleration at various fields of development is needed by embracing the foundation of UNS, Benteng Pancasila. Therefore, 5 strategic policy pillars were decided.
Halo Sasti.
Halo Mbak Lisa.
Makasih banget loh ya. Ini kita udah mau jam ini. Sebentar aku cek sebentar ya Bu Ratna. Bu Ratna?
Ya, bisa
oh,
didengar.
Oh ya, bisa Bu. Kita bisa mulai ya Bu ya. Ini semua sudah hadir. Bu Ratna?
Ya,
ya.
bisa didengar.
Ya, terima kasih. Sudah ada 52 yang ini. Oke. Okay. Good afternoon, everyone. Dear, I think we can start now. Now we have 54 participants in this room, but I think more are coming soon. Okay, so I would like to start. Good afternoon, everyone. Dear respected Head of Department of Biology, Dr. Sastia, distinguished guests, Dr. Matthew Chang from National University of Singapore, and Dr. Sastia Prama Putri from Osaka University. all academic staffs and students of Faculty of Mathematics and Natural Science, and to everyone, welcome to the guest lecture program. I am Elisa from this department, and today I will be the MC of this program. So this session will be divided into two, in which Dr. Sastia will deliver her first talk and then continue with Dr. Matthew Chang. And we will start our program with welcome speech from the head of Department of Biology. Welcome to Dr. Ratna Chanekse.
Thank you to Dr. Elisa. Good afternoon, everyone. Glad to meet all of you at this general lecture. Welcome to Dr. Sastia Prama Putri from Osaka University, Japan, and Dr. Matthew Wook Chang from National University of Singapore. Hi. Yeah, thank you a lot for being to be guest lecturer and sharing knowledge with us. I also express my gratitude to Universitas Sebelas Maret that's funding this event. Also, thank to Dr. Elisa Herawati and students who have organized the general lecture. And to all participants, thank you for joining our event. Let's improve our knowledge and enjoy this time. Thank you. I return to Dr. Elisa.
Okay, thank you very much for allowing some time to give the speech, Ratna. Okay, for this guest lecture program has been our routine event in our department. So for you who might still remember, previously we had our guests from Sweden University of Agriculture and also from Brunei Darussalam University. And today we are very excited to have our special guests from Singapore and Japan. So thank you for being here, Professor Chang and also Dr. Sastia. Thank you very much. Yeah. As I mentioned earlier that Dr. Sastia will deliver her talk first. So before that, I would like to briefly introduce her to everyone in this room. Dr. Sastia is a tenured assistant professor at Graduate School of Engineering, Osaka University. She received her PhD from the International Center for Biotechnology, Osaka University, in which she worked on the discovery of novel bioactive compounds from natural products. Her current research specializes on the application of metabolomics for synthetic biology as well as for food science and food technology, also focusing on food authentication and quality evaluation. She has awarded the L'Oreal for Women in Science for her work in copy luwak authentication. And she is the founding chair of the Early Career Member Network of the International Metabolomics Society and currently a member of the board of directors of the International Metabolomics Society. So Dr. Sastia will give a talk on mass spectrometry-based metabolomics application for microbial bioproduction. So Dr. Sasti, your presentation time is 20 minutes. Then we will continue with discussion for 10 minutes. Okay, please.
Hello, Melissa. Thank you so much for your warm welcome. Ibu Ratna, good to see you again. And Prof. Chang, good to see you in this virtual space. I'm really excited to deliver a talk again for UNS. UNS and US is very similar, just the swap, the abbreviation. So actually, earlier this year in March, I visited UNS. But at that time, I think it was not for the Department of Biology, but I think it was for food technology or agriculture. And the material that I brought to the audience was also different. It was about the quality improvement of agricultural product. So this time, I will be giving you a material that's quite different, and it's usually not the material that I share to the Indonesian audience. But because we have Professor Chang, who's an expert in synthetic biology, I'm really honored to share this session with him. And then we'll tell you a little bit about what I've been doing in Osaka University for the past, I think, 10 years that I'm working in the field of metabolomics. So I'm originally a trained microbiologist. And in fact, Elisa is was my classmate and my lab mate. So we've been friends for a long time. And I think perhaps this is the first time to give my Elisa a lecture about what I'm doing in metabolomics. I think for those who are in attendance today are probably very familiar with the central dogma in molecular biology. And you know well that the metabolome is situated very close to downstream, is a result of genetic expression. And it's therefore very close to the phenotype and function. The challenge is when we talk about the omics science is we would like to do a total profiling of a biological entity. And when we talk about the metabolome, of course, we would like to also do a total profiling of these metabolites within the biological system, which is not always easy because when we compare it with like DNA, it's quite easy to use like a single platform, and then you can do like using a next gen sequencing. Now you can sequence the whole genome within a shorter time frame. But with the metabolites, given the various types of metabolites and the diversity of these metabolites and differences in the physicochemical properties, it's still quite difficult to achieve this total profiling of metabolites. But nevertheless, we try. So what is actually metabolomics? It's actually a very interdisciplinary research between bioscience. So I'm a trained biologist and specializing in microbiology. So that kind of knowledge is really important to be able to design the experiment and to do the data interpretation well. But also because what we're doing is the science of measuring these metabolites, we need advanced analytical instrumentation. Then these advanced analytical instrumentation requires the knowledge of analytical chemistry. And in order for us to bring out the metabolites from inside the cell to outside of the cell, we need organic chemistry knowledge. So analytical chemistry and organic chemistry is also a very important component in metabolomics. Finally, when we talk about the omics science, it's very natural for us to generate big data. So this big data is a very complex data generated from this type of omics science. And it's very difficult to actually find the biological meaning within this big data if you don't use the tools like bioinformatics. And in that sense, we need also advanced statistics to be able to visualize the data in a way so that we could do this data mining and then finally interpret the data. So how is this metabolomics relevant to biotechnology, especially about metabolic engineering and synthetic biology? Professor Chang is also a good colleague of mine who's been working extensively in the field of synthetic biology, and he's one of the notable scientists in this area. And of course, everyone in the synthetic biology or metabolic engineering always aims to continuously optimize these bioproduction processes. And in that case, many different kinds of phenotypes are necessary to be improved. For example, like growth rate, production rate, yield, many different things might be very relevant. And these phenotypes affecting production is not sometimes easy to achieve because, for example, like these things like tolerance, it involves multiple pathways and interaction between pathways. And sometimes optimization of a single gene or like just simple deletion or overexpression result in something unexpected. So for example, like this kind of metabolomics driven strain improvement and in what way it can help is in the common approach, scientists usually use a rational selection. When you know the biochemical pathways and the enzymes governing these pathways, based on this genetic knowledge, you could construct the strain. For example, like trying to maximize the influx towards the pathway that we desire in order for us to achieve the desired production. Of course, increased production, faster productivity, and also more efficient use of the substrate. But sometimes, and many times, according to our experience as a molecular biology, when we delete a gene, something not expected happen because we're dealing with a biological system. It's not as simple as like a one type circuit. You block something and then something else go berserk, and then like you try to direct the flux towards something, and then some other byproducts is produced. And this result in unexpected cellular behavior. This often happens. How metabolomics-driven approach can help is to provide a deeper understanding of the metabolic state of host strains. Because when we want to produce something, we would like to know what is coming in between. Not only the input and then the output, we want to know what happens in the intermediate. And then if you can find a bottleneck reaction, or that if some reaction doesn't work optimally, then you can directly target that particular enzyme as the target for your strain improvement. In that sense, you can have a much more efficient process of strain improvement. So, of course, in the omics comparison, we always try to compare, like in the genomics level, genes to genes between strain 1 and strain 2. And then once you come up with some differences, you can highlight these differences and then probably generate some strategy for strain improvement. So based on this omics level comparison between strains, you could have some hints as to which genes, proteins may be important. And the good point about it is sometimes you don't need to understand everything in order for you to come up with a good strategy. Sometimes you just need to know enough for you to come up with a possible solution. So this is made possible by the development of high throughput omics technology. So most of these comparative studies have mostly focused on the genome or transcriptome. But as we already explained before, these levels are subject to downstream regulation and may not correlate to the final outcome. So the metabolome is situated downstream in the flow of genetic information and have more direct association with the phenotype. So if you have a complicated phenotype, for example, like involving tolerance, that you can't really pinpoint what kind of pathway is involved and might be a result of multiple pathways interacting with each other, metabolomics might be a good answer. You can subject these strains with different phenotype, and then you can measure these metabolite levels. Then please imagine if you have like strain A to G, and then each strain has different phenotype, and each strain will have different metabolites and different metabolite level. You can imagine in this metabolome matrix that the metabolome matrix will be quite complicated, and it's not possible for you to dissect it by your eyes one by one to know which one is important. So therefore, we need to do this kind of data mining by multivariate analysis. Then that kind of data mining will tell you what strains have the most similar metabolite fingerprint as a whole. Then when they are most similar, usually they will be clustered or situated very closely to each other. And where if they are very different, then they are going to be clustered far away from each other. So determined by the variance between samples. Then once you have all of these metabolome matrix, the metabolome information can be used to explain a particular phenotype. In this case, we can create a regression model in which the metabolome information acts as an explanatory variable, and the phenotype you would like to explain with metabolomics can act as a response variable, Y variable. Then you can make a prediction model. Once you make a prediction model, then you can highlight which metabolite is important for what phenotype. So this kind of concept is very relevant to many different kinds of life sciences application. It's not only very useful for the field of metabolic engineering, but as I mentioned before, I also deal with a lot of food and agricultural research, clinical research, and also basic science for basic biology to understand the function of some genes or understanding of the biological pathway. And for example, like in one of our publications that I will be explaining to you about, so I will be telling you many different kinds of application that we could achieve using this metabolomics. So this is in the sense of the biological bioproduction. Usually, of course, based on the expert knowledge, like Professor Chang is an expert, he has a lot of knowledge, and then therefore he could have a very good biological intuition and then come up with a hypothesis how to construct a strain. But even with the most expert, if we're dealing with an organism that is not like a model organism that we don't know much about, still, even in E. coli, we still have some surprises, like decrease in viability, overaccumulation of other byproducts that we don't want. And then afterwards, even experts have to go for trial and error. Then for metabolomics different approach, we can gain insight into the effects of gene manipulation and then facilitate metabolic engineering. So these are some of the papers that we have published in the past 5 years, starting from how we improve 1-butanol tolerance, how we can elucidate new pathways, and this was published in Nature Chemical Biology, or if we can actually improve the strain, and we have explained, have exemplified this strain improvement based on metabolomic strategy in several papers in recent years. So these are just the summaries of the case studies. And because today I only have 20 minutes, so I will not talk a lot about many of these different examples. Maybe in another time, if I have longer time, I can explain more of the examples. But today I'm just going to choose one example that I think would be best example to illustrate how we could improve bioproduction system using metabolomics. So this is what I'm going to talk to you about. It's about a cyanobacteria, which is a photosynthetic bioproduction system. The reason why I choose it, it's really cool because we can make bacteria produce biofuel only using sunlight and carbon dioxide. We reduce pollution and we use a carbon source that is free, of course, and we want to reduce it, it's carbon dioxide, and then we use light source from the sun that is also free. So this is something that, of course, everyone would want because it's a very important model system to convert atmospheric carbon dioxide to fuels and chemicals. And several papers have already started to demonstrate that from the past decade. So this is the key advantage of using cyanobacteria as cell factory over other model organism. As I mentioned, it uses sunlight as energy source and carbon dioxide as carbon source. And it's relatively faster if you compare it to algae or plants, but also it has the photosynthetic capability. And it has been known that it can engineer to produce valuable products. Then this is where something gets complicated. Microbial 1-butanol production is produced using CoA-dependent pathway available naturally by Clostridia species. As you may know, if you're a microbiologist, Clostridia is an obligate anaerobe, which means that it cannot operate with oxygen. Then you know that in photosynthetic bacteria, of course, oxygen is something that you can't prevent from coming. In the photosynthetic mechanism, then there's bound to have oxygen. So Liao Group, our collaborator from UCLA and who is now the director of Academia Sinica in Taiwan, he was the first to demonstrate the production of biobutanol production using E. coli. And with E. coli, there are not so many differences because E. coli also can operate under anaerobic condition, and also it's quite similar with cyanobacteria, Clostridia compared to cyanobacteria. But when it was put into a photosynthetic bacteria, almost no production, because in photosynthetic mechanism, NADPH is very abundant, whereas the enzymes from Clostridia mostly use NADH-dependent reaction. So the team switched the NADH-dependent reaction to NADPH-dependent reaction and yielded a small amount of production. Then they found that there's a very key enzyme downstream called BLDH, which is oxygen sensitive. And it is a bottleneck of the reaction. And they replace it with PDUP, which is an oxygen tolerant enzyme here, highlighted in red. BLDH is replaced with PDUP, and production increased so much. This was published in, the first paper was published in PNAS, and then this paper was published in EES. I think impact factor was 11, but it was simply just because they found a key enzyme and then they could increase the production so much. But what we found after was even more surprising. So we did metabolome analysis and also kinetic profiling of the CoA pathway metabolites. And we found that, in fact, butanol CoA which is supposed to be decreased because it has been replaced with an NADPH oxygen tolerant, NADH oxygen tolerant, is almost the same. So when we compared, this green bar is the level of butanol-CoA. And in butanol SE, in which PDUP enzyme has been changed, why butanol-CoA does not change? Then we reported this to UCLA and they say, maybe you guys make a mistake because we already fixed that reaction. So that reaction should now go very well. And we can see that from 1-butanol production being produced 7 times higher. We said, no, we replace this, we do this 3 times and we continuously get the same result. And this is absolute quantification. We are sure of the result. So this brings us to a very big puzzle, why this happened. So as I mentioned, sometimes unexpected things happen, even though we have a very good expert knowledge on the pathway. Then we found in the kinetic and metabolic turnover, we found that actually acetyl-CoA, which is situated upstream of butanol-CoA, is also increased. Why? We don't know. And then we checked that the synthesis rate of acetyl-CoA and butanol-CoA is also increased. So we think that something happened that caused more efficient butanol-CoA reduction, and it also enhanced acetyl-CoA synthesis. So this was a new finding. We published this in Metabolites, and we proved that this is not due to the insufficient NAD+ pool. And finally, we concluded that this is the mechanism, how it happened. When PDUP was optimized, actually, this reaction released free CoA. And without knowing, pyruvate to acetyl-CoA is actually another bottleneck reaction that Liao group has been working on for 4 years. 2 PhD students, nobody could solve the problem. Accidentally, we found out that by releasing CoA and then the use of free CoA to the pyruvate to acetyl-CoA, it promotes CoA regeneration. And unknowingly, when they fix PDUP enzyme, they also fix the bottleneck upstream from pyruvate to acetyl-CoA. This was found using metabolomics. So now we know that CoA regeneration is very important in 1-butanol production. So using that information, we know that we still have available butanol-CoA, and we improve PDUP even more by improving the enzymatic capability. Then increase the production for 74% further. Finally, we also expanded the target metabolite, not only focusing only on the CoA, but we also focus on other metabolites in the pathway. So we annotated 74 metabolites in the central metabolism, and then we compare the new strain with improved PDUP called DC7 and the original PDUP strain called butanol SE. And interestingly, we found that this strain also has another increased synthesis of acetyl-CoA. So if we make a diagram, this improved PDUP also improve the CoA regeneration. The more you improve the downstream reaction, butanol-CoA to butanol, the more you release CoA, and this promotes CoA regeneration. And this CoA regeneration, in turn, produces more and more acetyl-CoA. But if acetyl-CoA to malonyl-CoA is not condensed well, then acetyl-CoA goes to citrate, to TCA cycle. Then we found that the TCA cycle is in fact enhanced due to the abundance of acetyl-CoA that cannot be directed to the 1-butanol pathway. So what we wanted to do next is to improve this reaction by increasing the capacity of ACCase enzyme, which catalyze acetyl-CoA to malonyl-CoA. And as a result, DC11, a new strain, can reach the highest production with the highest productivity in the world right now. So it reached a production titer of about 420 milligram per liter in 6 days, while the original strain DC7 can reach a similar titer in 12 days. So we cut the production time to half time shorter, from 12 to 6 days, achieving the most efficient 1-butanol production system in cyanobacteria. So this improvement of 1-butanol-CoA to butanol resulted in a efficient increase, then we optimize a newly identified target for strain improvement, and the highest reported 1-butanol titer and productivity for engineered Xenococcus elongatus was successfully achieved. So I hope this could illustrate how metabolomics-driven approach is useful for developing strategies for optimization of pathway performance in microbial chemical production. And of course, it's not only for this particular strain. We have already demonstrated that the paradigm explored in this study could be expanded upon by targeting phenotypes for improvement in different industrial microorganisms. And of course, you can use many different kinds of platform. In this case, we use LCMS, but at times we also use GCMS, and you can employ alternate means of data analysis. I would like to close this talk by thanking Professor Fukusaki, Professor James Liao and his team, and all microbe team members of Fukusaki Lab. This research was funded under the JST NSF JST SciCorp funding from 2012 to 2017. Thank you very much.
Thank you very much, Sastia. Very interesting. And it's how long do you work to get all of this story?
Well, this is a long time. Actually, this work involved the first demonstration on how to construct up to butanol SE involves one PhD student from UCLA to work on that. And then when we found that CoA regeneration is important, that was a work of a master's student, Japanese student. And then how to improve the 2 cycles is a work of 5 years from an Indonesian student who is Mega, my student. So 5 years. In total, 7 years in our part.
Oh.
And it resulted in 5 publications in Q1 journals. So yeah, it's a long in 20 minutes.
Before we move to the discussion, question and answer. So I know that you will have another presentation after this, right?
Yes.
Yeah. Maybe it's better if we have the group photo session first. Okay.
Yes.
Okay, for everyone, if you don't mind, could you please turn on your camera, because we will have a photo session. Semuanya, tolong dibuka kameranya.
Oh, there's Matthew there. Hi, Matthew.
Good to see you. I really enjoyed your talk.
Yeah, thank you so much. I'm really excited to view yours too.
And I hope everything looks okay and all is well with your family. Okay, good.
Yes, great. Can't wait to visit you in Singapore one of these days soon. After COVID.
Yes. We've been waiting.
Yes, I think it's been a long time you haven't visited us in Japan too.
Yes.
Okay, I think everyone is ready. We have 3 frames here, so we will start from the first frame. Please hold about 3 seconds. 1, 2, 3. Okay, next. 1, 2, 3. Okay, one more. 1, 2, 3. Okay, thank you very much. Okay, so if anyone has a question, so could you please just turn on your microphone and can ask directly to Dr. Sastia? Ada yang mau bertanya? You can also ask in Indonesian if you hesitate to use English.
Ya, bisa bahasa Indonesia juga ya.
Yes.
Lancar.
Okay, Sasti. Okay, I think Yuda, you want to ask directly or okay? I think I have to read this from the chat box. There is one question from Yuda. How the metabolomics application for increasing food productivity, how the research method works, what molecules will be used, and how to apply metabolomics concept in daily life. Yeah, that's
Okay, thank you very much, Yuda. Nice to meet you. About metabolomics application for increasing food productivity. Okay, so similar with what I mentioned before, the concept of increasing a particular phenotype, Actually, if you remember that you can correlate metabolomics with any different kind of quantitative phenotype. So in my case, I use 1-butanol production, like titer, and productivity. You can also use growth. So in that sense, if you have crops or plants, then you use the productivity like growth or biomass, then you can use that as a response variable. Then if you have many different kinds of plants, same plant, but they have different phenotype, then you could have a prediction model that correlate the metabolites with that phenotype. So for example, if you have a plant that grows well and a plant that grows poor, what makes this plant grows well? What kind of metabolic pathway is enhanced in this good growth plant? Once you come up with the reason, once you know what kind of metabolite is relevant, you can try to target this metabolic pathway and find out the factors that can stimulate this metabolic pathway. Then in later on, you will be able to increase the food productivity. So the key is metabolomics can be used to explain any different kind of quantitative phenotype that you choose. But you have to make sure that you have good samples to compare it with. You have to have like a comparison in order for you to come up with the good metabolite to target. Then what kind of molecules will be used? The more you use, the better. So based on the knowledge of, for example, plant science, plant biology, you have to have a kind of hypothesis. What kind of metabolic pathway or what kind of metabolites might be related to your phenotype? So if, for example, in the plant biology, you suspect it's related to plant growth is related to, for example, assimilation of carbon source or like that, then you want to make sure that the metabolites that you target can cover those kind of metabolic pathway. Then number 2, how to apply metabolomics concept in daily life. Okay, I give you a very good example. Do you like coffee? I think everybody likes coffee. Who doesn't? Or some people don't like. But you know that Indonesia is very rich in the coffee, right? We have coffee from Toraja, from Mandailing, from Java. But actually, the coffee bean is the same. Maybe it's result from the same species. But when you grow it in different places, it results in different type of coffee. And this is due to the microclimate, the elements in the soil and the temperature, so many different things in that area, the geographical climate, that result in different kind of coffee. Then we can tell what is special between these different kind of coffee. For example, what is different between Arabica and Robusta? Why does Robusta coffee feel bitter compared to Arabica? It turns out because it has higher caffeine content and caffeine tastes bitter. And you can explain. And for example, we do, for example, food quality. We have different kind of tea that always wins competition in Japan, like green tea competition. Then all of the good green tea always have something in common. It always has the sugar content low and epicatechin or the other flavonoids high. Then you know if you want to have a high quality tea, this is the type of characteristic you need to have in a good quality tea. So in that sense, we can guide this kind of food quality improvement once we know what we need to improve. Otherwise, we are just going to do it randomly, like try many different things until we can get something that is good. So metabolomics is basically to make the whole process much more efficient.
Okay, Yuda, I hope you satisfied and understand the explanation. How about any other else? Any questions? from other students or from staffs, maybe? Okay, Sasti, I have a general question. This is a general question about your work. Okay, so it seems that your work is more to metabolite profiling, not metabolite fingerprinting.
Yes.
That's a different thing, right?
Yes.
Okay, so in the case of metabolomics, because it aims at comprehensive and quantitative analysis of wide areas of metabolites in biological samples. So between metabolite profiling and fingerprinting, which one is more true omics?
Okay. Thank you for your question. It's an excellent question and fundamental one. Actually, metabolic fingerprinting is when you try to do the total profiling and take everything into account as a fingerprint. So those peaks you have, you can analyze them all and then take everything into account and make it into a pattern. But you don't need to do metabolite identification. You don't need to know what these metabolites are. But metabolite profiling, you need to annotate them. And then based on the ones that you annotate, you can also include the ones that are not annotated, but somehow there's a kind of like a subset of all of the peaks that appear. Metabolite profiling will certainly have less number of features, chemical features, because you need to have the process of metabolite annotation. Then the true omics is actually metabolite fingerprinting, right? Because if you want to do a total profiling, but there is a limitation in that sense. If you're a biologist and if you want to do metabolite profiling, you can do that if you want to discriminate two things. Just for example, yes or no, good or bad, fake or authentic. If you just want to do complete discrimination without knowing what caused the discriminating factor, it's okay to do metabolite fingerprinting. But we are scientists. We need to know, and so that we can discuss and generate new strategies. So that we want to have a biological information on what kind of pathways involved, what kind of metabolite is responsible in order for us to do further research. So in the case of application in the industry, whereas you don't really need to explain the science, you just need to achieve the purpose of discrimination. Metabolite fingerprinting is very good because it's cheap, it's rapid, it can use like FTNIR, for example, that is abundant in Indonesia. You don't need to use mass spectrometry. But metabolite profiling is good for science. If you want to like dissect the pathway, reconstruct the phenotype, understand the meaning and the importance and contribution of a particular metabolite to the phenotype, then you need metabolite annotation.
Thank you. Thank you for your answers. We don't have much time anymore. Is it time limit? Okay, you have to go. Okay.
But
thank you
I'm
very much.
happy to answer questions by email if there's someone who still have questions but actually shy. I'll be happy to answer them after the session, and then I'll be joining again after Professor Chang's talk. After I give my
thank you very much. Okay, see you again. Thank you, Dr. Sasti.
Okay, thank you very much.
Bye. Bye for now. So we would like to proceed to our second special guest. We have here Professor Chang. Thank you very much once again for allowing some time to be here. And I really appreciate your, you allow us to switch the presentation order in the last minutes. Thank you very much. So
thank you.
Professor Chang, how are you doing?
Very good. Thank you so much for having me here today.
Before we start, I would also like to briefly introduce our second guest. Professor Chang is an associate professor from Department of Biomedical Engineering, National University of Singapore. He is the director of Wilmer NUS Corporate Laboratory, director of Singapore Consortium for Synthetic Biology, and director of NUS Synthetic Biology for Clinical and Technological Innovation. His research interest is reprogramming biological functionalities for energy, healthcare, and environmental application. He published many articles in highly reputable journals, and just to name a few, for example, the synthetic yeast genome reveals its versatility, published in Nature 2018, and engineered commensal microbes for diet-mediated colorectal cancer chemo prevention, that is published in Nature Biomedical Engineering. Wow, awesome. Okay, Professor Chang, so your time is 20 minutes, so please, yeah, the time is yours. Thank you.
Okay, thank you so much for the invitation, and it's a great pleasure to be here today. And hopefully everybody can see my screen. Okay.
Yes, we can see it clearly.
Okay, that's great. So I understand that there are many students in the audience. So it kind of tailored my presentation to the students who might be the major the audience for the presentation today. So what I'm gonna actually talk about today is why biotechnology is going through some changes, and why I believe by engineering biology, actually, we can solve some of the societal issues that face our world and our region. So again, my name is Matthew Chang, and I'm from the National University of Singapore. As you may know, biology is everywhere. If you look around, you can easily find biology and also the product of biology. Nature, trees and flowers, birds and animals, and even ourselves are in fact a product of biology. So to me, biology is truly amazing and really awesome. And if you think about our body, and many actually can come to the realization that we are not actually alone. And our body actually houses numerous microbes in our body, and they actually help with our digestion and also the metabolism. In fact, in terms of cell numbers, the microbes actually make up about 40% of our body. The use and application of biology is called biotechnology, and as you know, biotechnology actually has been around for thousands of years already, impacting various aspects and every corner of our life. So of course, some food products, including bread and cheese, are made through biotechnology. And then actually been made and manufactured for thousands of years by the power of biology. At the same time, a wide range of medicines are currently being produced by biotechnology as well. So what actually makes biotechnology very exciting and interesting? And I believe there are a few characteristics of biology that actually make biology and biotechnology extremely exciting, and that we can capitalize on to develop the next generation biotechnology. So as you know, biology is self-replicating. I mean, cells can reproduce themselves in an autonomous manner. And this is truly amazing, an amazing aspect of biology. At the same time, I really believe that the fact that biology is self-replicating can be harnessed for a range of biotechnology applications. And at the same time, biology actually comprises many different types of molecular machinery. I mean, this short animation actually shows how DNA is replicated inside the cells, which takes place numerous times at any given time. And this process, DNA replication, as you know, involves molecular machinery that consists of a group of enzymes and DNA fragment. And this process takes place in a highly precise manner with extremely low error rate as well. And these images are E. coli microbes, which all of us actually have in our body. And the cells are about 1 micrometer long. This actually shows a scientific illustration of E. coli cells. And this circle region indicates a flagella, a molecular machinery that enables E. coli cells to swim and move around. But what is amazing is if you take a photo of the flagella using electron microscope, you see an amazing molecular machinery that is made up with 25 different proteins. And these 25 different proteins are precisely produced and arranged as shown in this figure, spanning about 40 nanometers. We now actually talk about nanotechnology as a future technology, but as I mentioned before, biology has already mastered nanotechnology long time ago. And this is the foundation of biology, the DNA, RNA, and protein, and their transformation take place through the same process called central dogma, which means if we reprogram DNA cores, we can actually change the behavior of the cells. And another amazing feature is that all the living cells actually share very similar metabolism mechanisms. That's why the metabolomic approaches that Dr. Putri just mentioned is becoming extremely relevant and important in the engineering of biology. And actually this image shows the core metabolic pathways, even though they look extremely complicated and complex, all the living organisms actually share very similar metabolic pathways at the core of their cells. And computer codes consist of 0 and 1. By using different combinations of 0 and 1, I'll argue that we can program computer software, and in the same manner, by changing the biological codes, which consist of A, C, T, G, shown on this slide, we can reprogram the behavior of the cells by changing the order of A, T, G, C. By changing the combination of these genetic codes, we can also reprogram cells. And this is a genetic code of insulin, which we produce to modulate the sugar level in the body. And we can make the DNA that shows the same genetic code, and we can put that into microbes, and we can reprogram the microbes to produce the same hormone insulin, and which you see on the market as a diabetes drug. The fact that biology is replicating and programmable allows biology to be able to benefit from other scientific disciplines. And now is a very good time to be a biologist and biological engineer in my view, because many interesting scientific disciplines such as medicine, chemistry, engineering, computing, and even robotics are now converging into biotechnology. This convergent science, outcore synthetic biology or engineering biology. And synthetic biology, briefly mentioned, aims to develop new biological products by engineering of biology. And one of the reasons I'll argue synthetic biology possible now is that our ability to read and write genetic codes of biology has exponentially increased for past decades. As I mentioned before, we read and we write the genetic codes to reprogram the operation of the computer. In a similar manner, now the advancement of biotechnology and biology has enabled us to read and write DNA in an extremely efficient manner so that we can reprogram the living cells as well. So systems biology approaches, including the metabolomics that Dr. Putri have just briefly illustrated, can really help us better understand how the living cells really work and operate. And based on this understanding, and by changing the genetic codes at the bottom of the central dogma, actually we can reprogram the living cells. About 20 years ago, the sequencing of the human genome took about 10 years and cost about $300 million in US dollars. But now the entire human genome can be sequenced within hours for about $1,000. So as a result, we now have a genetic codes for every major organism on the planet. And all these genetic codes can be reprogrammed to add new biological functions to the cells. At the same time, the biotechnology also has enabled us to directly synthesize DNA genetic codes and introduce them into the cells. We can now directly synthesize very long DNA fragments. And then the price of DNA synthesis has dropped over 100-fold for past a few decades. And this advancement in DNA writing technology has allowed the design and construction of even genomes from the scratch. So my lab is part of the international consortium on this synthetic genome project. And now we actually embark on a very ambitious journey aiming to redesign and construct the 16 chromosomes, all the chromosomes of Saccharomyces cerevisiae, which is an yeast, the cell, which actually has been used for thousands of years for many types of biotechnological applications. We actually teamed up the many universities around the world, and then each member and each institution has been redesigning and constructing each of the chromosomes of Saccharomyces cerevisiae. Now, I think within the next 6 to 8 months, we can really generate synthetic chromosomes which can replace the native chromosomes of Saccharomyces cerevisiae, which can mark the world's first eukaryotic synthetic genome. Another exciting development that I just want to briefly touch upon is the expansion of genetic engineering and bioengineering tools. And these tools enable precision engineering of DNA and cells, and this type of bioengineering tools include CRISPR and some other genetic engineering toolkits. As I mentioned before, various disciplines are converging in biotechnology, and especially engineering framework, which consists of design, build, test, learn cycle, is being implemented in various biotechnology projects to make the engineering of biology faster, cheaper, and more systematic. And there are tremendous efforts being made to make the engineering of biology cheaper, faster, and more systematic. And one of the approaches that many labs around the world, including ours, are employing is to automate some of the key steps involved in biological engineering through robotic systems and also the artificial intelligence algorithms. So biology, in my view, is increasingly becoming automated, which can increase the speed, accuracy, and reproducibility of the process. And automation empowered by the robotic systems you see here can actually really bring down the cost associated with the engineering of biology. We also co-founded the Global Biofoundry Alliance, Now, this alliance actually has 26 members from all over the world, and we are now developing and collaborating to solve some of the pressing issues facing our society. So this convergence of various scientific disciplines, in my view, is synthetic biology. So the convergence of biology, medicine, chemistry, engineering, and computing in robotics. And that's why I hope to argue that synthetic biology can really change the way we live, and also we manufacture and we treat human diseases. And I would argue that we might be seeing the beginning of a biotechnology industrial revolution. And the reason is that if you look around, actually, most of the materials actually we see and we touch around us actually originate from petroleum. So carbon feedstocks, including petroleum, are converted to commodity chemicals and materials through synthetic chemistry. But I'd argue that through synthetic biology, as Dr. Putri mentioned briefly, we can also produce a range of commodity chemicals from biomass feedstocks in a much more environmentally and friendly and sustainable manner. This can actually make a significant contribution to the sustainable manufacturing with much smaller carbon footprint and much less waste. So bioeconomy is an economic activity fueled by biological innovation, and I believe that synthetic biology can hold key to developing sustainable bioeconomy and future through biological innovation. So even though the synthetic biology is still a young field, actually has shown a lot of potential to change the way we manufacture and treat human diseases. So I just want to briefly touch upon some of the applications of synthetic biology in the coming slides. So synthetic biology can actually make an impact on the chemicals and materials industry and production. And as Dr. Putri mentioned, some of the examples include the production of the key building block chemicals and also materials and even biofuels using microbes. So microbes can be engineered, as you can see here, to produce chemical building blocks of polymers in an environmentally friendly manner from renewable bio-based feedstocks. And very recently, the US Navy has developed a new fiber-resistant composite materials using engineered microbes for aerospace and other unmet advanced materials needs present in the defense space. And even consumer products, we can envision producing through synthetic biology. One example is DNA storage, and all our data can be digitally stored in a computer hard disks and flash memory chips. But DNA can also store the same type of data at much higher density with much less power and much longer retention time. In fact, 1 kilogram of DNA can contain, according to the literature, can contain the world's entire data. And then, Microsoft, the US company, has been running DNA storage program since 2015. And then the last, industry sector that I believe that synthetic biology is making an impact on is medical industry. Now, actually, we have demonstrated that the microbes can be engineered to produce novel synthetic medicines, including the anti-malaria drug as shown on this slide. And then even human, the defense cells actually can be modified and engineered, and then they can actually be put back to the patient so that patients can attack the target cancer cells in vivo, and which is now really revolutionizing the way we treat cancer cells. And in our lab, we've been engineering and we've been using the microbes living in our body for disease treatment and prevention. And then we actually have shown that using our own microbes, we can show very targeted therapeutic functionality, including anti-infection, anti-cancer, and also anti-metabolic disease. And actually in animal models, actually, we have shown the efficacy against target infectious disease. And also, we also showed very high efficacy against colon cancer in animal models using the microbes that live in our body. So the message that I actually really hope to convey to you today through this presentation is that synthetic biology is a very young field, but has shown great potential to make a significant impact on various industries and many aspects of our life. And also synthetic biology is truly accelerating biological industrial revolution. In Singapore, one of the organizations I'm heading is Singapore Consortium for Synthetic Biology. We actually have many academic members and industry members, and we are collaborating very actively to bring a very tangible benefits of synthetic biology to the public in a more regionally. We actually formed Asian Synthetic Biology Association, which actually has more than 250 members across Asia, and also has more than 50 universities as academic institutions. And we've been meeting very frequently and interacting so that we can really accelerate and speed up the translation of synthetic biology research taking place in Asia. With that, I'd like to thank my students and my funding agencies, and I also want to thank you for your attention.
Yeah, thank you very much. That's awesome. So to us, synthetic biology is quite a new because not many of us are working in that area. So today we learn a lot from you. Thank you very much.
My pleasure.
Okay, is there any questions from the audience? Okay, Professor Chang, I'm interested in your slides about synthetic chromosomes that you showed.
Yes.
Yeah, synthetic chromosomes. So in my mind, that chromosome is a big thing, right? And speaking of synthesis of large DNA, I think the challenging part is the assembly step. Is it right? So because usually the contiguous DNA usually assemble from smaller, from the more easily synthesized DNA. So what is the strategy?
Right, yes, that's an excellent question. That's why I actually spent some time in my presentation emphasizing the importance of DNA synthesis technology. So this type of large-scale genome synthesis not be made possible without harnessing DNA synthesis technology. So what we have been doing is, in silico, we've been redesigning the entire chromosome. And then we actually added some new biological functionalities to the artificial chromosomes, so that eventually all the chromosomes can be harnessed and used for specific target biotechnological applications. And then, based on the redesigned chromosome layout, we chemically synthesize long DNA fragment, which usually contain about 10,000 base pairs. So 10,000 base pairs. And then in our lab, actually, we piece them together so that actually eventually the mega chunk we call, which actually consists of a few the large DNA fragment, each mega chunk actually can, using the homologous recombination technology, replaces the target region of the native chromosome. So one mega chunk at a time, actually, we are replacing the natural region of the chromosome with a synthetic and newly designed the DNA fragments. And then through this process, we in my lab, actually, we are responsible for reconstructing chromosome 15 of Saccharomyces cerevisiae. And chromosome 15 actually has more than 1 mega base pairs on the chromosome. So about 50,000 base pairs at a time, actually, we've been replacing the native chromosomes. Now, actually, we are seeing the finish line. And I think, in a month or so, actually, we can finish the reconstruction of chromosome 15 in Saccharomyces cerevisiae.
Wow. Okay, thank you. That's a very advanced technology, I think. Okay,
Yes, yes, quite advanced. Yes, I have to
yeah.
admit.
There's a question from one student, Rizka. Hello, my name is Rizka. I don't really understand about biology is programmable or how we can program DNA or gene. Can you please tell us in detail, maybe the methods in detail? How do you reprogram, redesign, fabricate, or make it like functioning as you want, something like that?
Right. I mean, that's an excellent question, and thank you so much, Liska. So what I meant by, my argument that biology is programmable, because biology, as you can see from this slide, actually consists of 4 genetic codes. So A to G, C, as everybody in this audience knows. So this actually dictates what proteins are expressed in the living cells, and also what metabolites are actually produced in the living cells as well. So if you change the genetic codes, you can also change the behavior of the cells as well. So one example I actually brought to you today is a just very simple example that has been around for several decades. It's actually production of insulin. So insulin actually our human body naturally produces, and this is actually genetic code of the insulin protein. We can synthesize the same DNA fragment, which actually shows the same DNA sequence. And then we can actually put the same DNA sequence into microbes, because microbes actually contain the same DNA codes as the one that leads to insulin. Therefore, we can reprogram the genetic machinery of microbes to produce insulin protein. So that's one example of reprogramming living cells for specific functionalities. And hopefully I answer your question, Rishka.
Okay, here's one question. Do you still have time, sir? Do you still have time?
Oh, me? Yes, of course.
Okay.
Yes.
Okay, because we got many questions here. I'm really interested in yours. This is from Ardo. I'm really interested with your statement was biology is programmable. Is there any chance to edit the genome of SARS-CoV-2 as therapeutic COVID-19? Yes. So.
I mean, that's an interesting idea and proposition. Yeah, maybe we can create, anti-COVID-19 agent through genome engineering. I mean, that's certainly possible if we can really precisely manner give specific functions to the living virus or microbes. So yes, that's a possibility, but I don't know whether any work related to this has been done, and I think people are still relying on more conventional and safer approaches for vaccine production and also antibody production. But as vaccine production usually takes a lot of time and usually takes several years, and the world record is actually about 4 years. But now we can probably, this COVID-19, and we're actually really benefiting from all the advancement made in biotechnology. That's why now we are actually seeing the possibility of being able to produce vaccine even within a year after the pandemic, the outbreak. So that's certainly, the power, example of the power of biology, which can be done onto various aspects of biotechnology, technology development.
Okay, thank you. This synthetic biology area reminds me to, actually it's also genetic engineering. Is it right?
Yes, I mean genetic engineering actually could be one tool, and also at the same time, I'd say actually genetic engineering, we try to manipulate a small set of genes, but now, as I mentioned to you before, we can now envision just rewriting the entire genome. So we can say that we can probably start envisioning even larger scale, engineering biology.
Okay, so
and technologies that they can benefit from that technology. Yes.
Okay, so in genetic engineering, maybe you deal only individual gene, but in synthetic, you are dealing with a standardized set of
Yeah,
DNA,
but
something like that.
yes, but of course, genetic engineering is the core of synthetic biology technology. But now, as I mentioned to you before, genetic engineering is also becoming automated, and the scale is becoming larger, so that effectively we can engineer, we can actually make the engineering of biology faster, cheaper, and more systematic.
Okay, we have two more questions, if you don't mind. I
Yes,
don't know
I don't mind,
why
yes.
these students hesitate to talk directly. They prefer writing on the chat.
No problem.
This is from Helen. Is there any limitation in choosing organism for synthetic biology research? Because
Yes,
your presentation
definitely.
talks more about microbes application.
Yes. So that's an excellent question. Even though actually I talked about the potential of synthetic biology, there are a lot of room for, entire field actually has a lot of room for improvement. And one of the limitations is the lack of genetic tools that we can tap onto when we engineer biology. That's one of the reasons that my talk actually has been more focused on microbial engineering, because we have a lot more tools, genetic tools, by which we can very efficiently engineer microbes. But mammalian cells and even plant cells, we have some tools, but usually this type of research endeavor takes a lot more time, requires a lot more resources. And that's why I actually placed more emphasis on the microbial elements of synthetic biology during my presentation today.
Okay, thank you. So this is the last question, I think, but I'm not really understand.
So I think the question is, Monica. Thank you for the question. And I think the question is whether the crop actually can be engineered to improve the productivity. The answer is yes. There are quite a few companies which are trying to use synthetic biology and genetic engineering approaches to improve the crop's productivity. So the answer is yes. And also now because of the climate change, we're actually hoping to make our crops actually become more tolerant and robust against climate changes. Yes, so there are having a lot of efforts actually being made to improve the crop productivity through engineering biology approaches.
Okay, thank you. I said the last question, but actually there is one more. Is that okay?
Oh, yes. No problem.
So
yes, my
this
pleasure.
is from Yuda. Do you deal with, he's asking about ethical clearance. Do you have to deal with some ethical clearance?
Of course. Yes, of course. So ethical clearance and also the regulatory clearance must be obtained before we deploy any type of products that are made through biological engineering and engineering biology. So that's something that entire community is paying a lot of attention to. So we actually are in very frequent discussion with the regulatory authorities about the eventual utility of some of the products that we develop through this type of approaches. Yes, the answer, we must obtain the regulatory and ethical clearance. And we can, as a scientist, of course, we really have to do our due diligence, ensuring, whatever products actually we develop are safe, and also don't lead any unexpected adverse impact on our society and also on our nature.
Okay, thank you very much. So we have a very great discussion today, but I think our time has reached the limit. So thank you very much, Professor Chang, for
Thank you.
sharing with us a very inspiring, and this is a new area even for most of us. This is
okay, great. Happy
thank
to
you
share.
very much. And
thank you.
Sasti, you have joined again. So okay.
I'm back.
Hi. So, are you both working on a project?
Yeah, we used to work on a project. Now it's on hold. Yeah, Prof. Chang.
Oh yes. We actually have to revisit.
Yes, we have to revisit
soon.
the project again.
Yes, definitely.
Okay. So, well, once again, thank you very much. So I hope on the other time, probably when it's the pandemic has over, we can have a chance to discuss face to face. Stay safe and stay healthy for both of you. Thank you for giving a very great talk today. And okay, I think that's all. So see you next time. Bye. Thank you.
Okay,
Thank you
take
so
care.
much for inviting us. Thank you for having me.
Thank you.
Thank you.
Bye-bye.
Sastia, yes, we can write to each other.
Yes,
so that we can keep
definitely.
the
I'm
conversation
very
going.
much
Yes.
looking forward to it.
Yes. Okay.
I'll
Bye.
be waiting for your contact. I'll
Yes.
contact you. Bye-bye.
Okay. Bye.
Terima kasih semuanya. Terima kasih yang sudah menyimak. Terima kasih. Ya, bye.